Hello, good morning to all my dear students. First of all, happy Diwali to all, and you are celebrating the best festival. Let us start from a new topic, which is based on sexual reproduction on flowering plant. Sexual reproduction of flowering plant. Yesterday, we had talked about the basic structure and morphological characteristics of flowering plants. Today, let us start from a new topic, which is pre-fertilization events in flowering plants. Pre-fertilization event. Student, first of all, we should know what are pre-fertilization events of sexual reproduction. Pre-fertilization events of sexual reproduction in which the fertilization, the process of fertilization or prior to fertilization. Fertilization is the process of fusion of male and female gametes, fusion of male and female gametes. This process is known as the fertilization, but pre-fertilization are the processes or events that occurs prior or before the fertilization process. So let us start from a microsporogenesis process in case of male reproductive organ in flower. As you already know about the structure of anther, or we can say the complete is the stamen. Stamen consists of two structure. First at the tip is known as the anther, and the stalk-like structure is the filament. As you know about the anther are the part of stamen in which the pollen grain are produced. Pollen grain are produced inside the anther. If we cut the transverse section of anther, we can see a structure is known as the microsporangium. If we cut the transverse sections of anther, we obtain the four sac-like structure. One is called the microsporangium, and all the four groups are known as the microsporangia. I am repeating again my topic. What I am trying to say, or what we are going to discuss in microsporogenesis. The topic name is the microsporogenesis. Sporogenesis. As you names just that, micro means very small, and sporo means spore-like structure, small spore-like structure. Genesis means, in case of complete biology, when we face this word is the genesis. Genesis means formation of any things, formation of new things. In case of microsporogenesis, here the formations of microspore. The process of formation of microspore from microspore mother cell is known as microsporogenesis. Sometime examiner may ask the definitions of microsporogenesis in CBSE board, and also in case of NEET exam. CBSE board, you can put a question, you can put from your question is microsporogenesis, or define a microsporogenesis process. You can write down in simple form the process of formation of microspore from microspore mother cell, and by the process of meiosis. Meiosis is a type of cell division in which the reduction of chromosome number if any cell is diploid or 2n, we can say the diploid cell after meiosis, the ploidy level of diploid will be haploid, or you can say that it becomes n number of chromosome. If the parent cell has 2n or diploid, after meiosis it can get converted into haploid cell. Let us start from a basic concept of the microsporangium, and after that we will study also about the complete process of microsporogenesis. First of all, this one is the sac-like structure or microsporangium, and the group is called the four groups of pollen sac microsporangia. Say for example, you all are aware by the bacterium or bacteria. Bacterium means simply a single bacteria, and bacteria means the group of bacteria or colony. We can say the colony is the group of bacteria. When we're talking about the bacterium, we consider only single bacteria. Bacteria means plural or group of bacteria or colony, and bacterium means the single bacteria. Let us start from microsporogenesis process. This is the sac-like structure, and microsporangia or microsporangium contain a sporogenous tissue. If we consider this one here, it consists of sporogenous tissue. How the sporogenous tissue is filled inside the microsporangium? This green marker indicates the sporogenous tissue which is present inside the microsporangium. Sporogenous tissue. And we know about the cell or group of cell. Group of cell is known as the tissue. If this is a tissue, we can consider it consists of different type of cells or similar type of cell. It having a cell which is known as single kind of cell, but group of cell which is known as the pollen mother cell or MMC, microspore mother cell. The full form of PMC is the pollen mother cell, and MMC stand for the microspore mother cell. What happens in case of microsporogenesis? This is the mother cell because it gives rise to produce a new types of microspore or haploid for spores. First of all, microsporangium converted into pollen sac. Pollen sac is the sac-like structure in which the pollen grain are produced. If by taking an example, so this is the pollen sac. Pollen sac is a sac-like structure in which the pollen grain are produced. Pollen grains. Sporogenous tissue consists of PMC and MMC. After meiosis, first of all, we taking an example of meiosis 1. Meiosis 1, we can say the reductional division. Reductional division. Here the ploidy of PMC or MMC 2n means diploid cell. After undergoing the meiosis 1, there is two phases of the meiosis process, meiosis 1 and meiosis 2. Here, first of all, we take an example of reduction divisions or meiosis 1. Meiosis 1 is the reductional division, whereas the meiosis 2 is equational division. First of all, the MMC or PMC undergoing the meiosis 1 to produce a conjugated form, but they are separated actually. Haploids all having the ploidy level is N, or in form of tetrad. Tetrad means there is formation of 4 different type of cell, but in conjugated form, but ploidy level is N, haploid, all having the ploidy level is haploid. We can say this one becomes a haploid nucleus of each. After that, meiosis 2. Meiosis 2 is the equational division. The basic concept is the meiosis. Meiosis 1 is the reductional division because the chromosome number becomes to half, 2n becomes n. But in case of mitosis, mitosis is the equational division, or mitosis, or you can say the meiosis 2. Meiosis 2 is also a type of equational division. Here the meiosis 1, but in case of when the tetrad cells converted into haploid or mature haploid spores or microspore, actually the chromosome number will same in case of daughter cell, because meiosis 2, we already discussed about the meiosis 2 is the equational division, or similar considered as a mitosis 1. After that, here is the formation of 4 different type of 4 microspore. 4 microspore. By taking an example, after meiosis 1, there is production of 4 haploid cells. 4 haploid microspore. All the processes are similar, but we can consider only single cell example. Here the new cell division occurs, which is mitosis. All mitoses are similar in the remaining 3 cells, but we can take an example of single cell because the events are similar in the remaining 3 cells. Mitosis produce, after mitosis, a microspore produce, or it will becomes haploid to haploid and 2 cell stage pollen grain. Microspore actually technically known as the pollen grain. Pollen grain or microspore, similar one. Unequal division. There is unequal division. Unequal means no related to chromosome. Unequal means division of cell. One is the smaller cell and other one is the larger cell. This one is the larger one and this one is the smaller. The larger cell is known as the vegetative cell and also known as the tube cell. Examiner may be asked from another name of the vegetative cell. Sometime question may be arise, what is tube cell or what is vegetative cells, but they are same things. You can write down the name of vegetative cell as tube cell or tube cell as vegetative cell, vice versa. The another cell is the smaller cell, or you can say the generative cell, which is plotting in the cytoplasm of the larger cell or vegetative cell, because we can say this is a very small cell as compared to the larger cell or tube cell. The generative cell plotting in the cytoplasmic content of the generative cell, this is the plotting cells and the cytoplasm of vegetative cell or tube cell. After that, here this complete pollen grain is surrounded by 2 layers of cell. One is the antenne, the inner cell, and outer one is the axine. and here is the same one, which is the two cell stage. One is the generative and other is the tube cell or vegetative cell. This one is the generative cell. Now this is known as the complete mature pollen, or you can say the mature microspore. Mature pollen. Any query, students? Any question regarding the concept of microsporogenesis? You can take a screenshot. Okay. After the formations of mature pollen, the two cell stages, one is the vegetative cell and another one, which is a smaller cell, is the generative cell. Vegetative cell, also known as the tube cell, and the two structure surrounded the pollens, or two layer, you can say. One is the entine, which is inner layer, this one is the outer layer, exine, and this one is the entine. Entine is the inner layer of the pollen grains, inner layer. Where the X means outside, or the outer layer of the pollen. Outer layer of the pollens. The question maybe arises, what is the constituents or what are the compartment present or substance present in entine? And question may be arise, exine is made up of dash. Sometime examiner may be such type of question. The exine of pollen made up of dash. Option will be sporopollenin. Option B may be cellulose. Option C, both A and B, and option D, all of the above. What will be the right one? The sporopollenin is the right answer of the exine, because exine is made up of the sporopollenin, which is resistant to high acidic environment or high temperature. It provides a strong mechanical support to the pollens, but there are some regions where the exine or this layer is absent in the mature pollen. By taking an examples of small pores, here we can say these are the small pores. Small pores where the exine is absent. These pores are known as the germ pores. And germ pores are the regions. Germ pores are the region from where the male gametes are released. Germ pores are the regions of exine where exine region, not exine region, where the exine is absent. There is small pores from which the male gametes are released. male gametes are releases, because the male gametes are formed inside the generative cell. Generative cell means it generates something. What it generates? It generates the two male gametes. It generates the two male gametes. So we can say, so named as the generative cell. And exine spores are known as the germ pore. From here the male gametes, the germ pores are the region or exine where the male gametes from which male gametes release outside the pollens. After that, the question maybe arises, what is microgametogenesis? Microgametogenesis. We have completed the one part which is microsporogenesis, because microsporogenesis is simply there is the formations of pollen grain or microspore. But the question may be asked by examiner, what is microgametogenesis? Here we have not discussed about the microgametogenesis till now, because the gametes formation process is known as the gametogenesis, but here is the process of formation of spore or sporogenesis. Microgametogenesis is the process in which the generative cell mitotically divides to produce two types of male gametes. When the generative cell, this one is the generative cell, it mitotically divides to produce two types of male gametes. Now the process is known as the microgametogenesis. Why the micro word? Because the size ranges is micrometer, 10 raised to power minus 6 meter. 1 micrometer is equals to 10 raised to power minus 6 meter. This is the size of micro. This is very small gametes. Mostly the female gametes are larger and the male gametes are very smaller or micro size. We can say the micro gametes. And gametogenesis process is the formation of gametes from generative cell. This one is the generative cell. Generative cell mitotically divide. Generative cell mitotically divide to male gametes. This is all about the microsporogenesis process or microgametogenesis process. Student, if you have any query or question regarding this topic, you can ask me freely. This topic is very important about the board exam or sometime NEET, because the questions maybe arises different types in case of NEET exam. Say for example, we had discussed about the exine which is made of sporopollen, but we had not discussed about the entine is made up of dash. Entine is made up of cellulose plus pectin. Sometime examiner may be put such type of question. The entine is made up of dash. Exine or entine. Entine is made up of this type of substance, cellulose or pectin. Cellulose, because the plant cell wall is made up of cellulose. Plant cell wall is made up of cellulose and some other hemicellulose are present. Now the question based on size of pollen. Size of pollen. It ranges from 25 to 30 micrometer. Now you can take a screenshot. This is all about the microsporogenesis process, which is a pre-fertilization events, as we have already discussed about in the starting of the lecture. What are the pre-fertilization events? This include now microsporogenesis. This include in pre-fertilization event. Pre-fertilization events, because these events is occurs prior to the fertilization. Fertilization means before the fertilization, but fertilization is the fusion of male and female gametes. Fertilization, we can say simply the definition of fertilization: the process of fusion of male and female gametes. Example: egg cell plus male gametes to produce zygote. Fertilization results to form zygote. Here is the N haploid cell fusion to produce a diploid cell 2N. Zygote is 2N. Single diploid cell zygote. There is basically three events: pre-fertilization, fertilization, and post-fertilization. Post means after the fertilization. Post fertilization. Pre means prior to fertilization. Fertilization means in which actually fertilization taking place. But the post fertilization are the events in which all the embryogenesis process or embryonic development. We can say such example. Say for example embryogenesis and embryonic development. Embryonic development include in the post-fertilization events. Here the gamete formation, pre-fertilization, gamete formation. Okay students, if you have any query about the complete topic which we had discussed now, you can ask me. Tomorrow we will start from a new topic, but also related to pre-fertilization, which is megasporogenesis. Mega means the female gametes, formation of large spores. Okay बच्चों.